Okay, I have noon right here in New Haven. Happy New Year, everybody. Welcome to a new series of our global immunotalks for this new year, 2021. I'm Carla Rothlin, and I'm here with one of the co-organizers, Elina Zuniga, who will introduce our fabulous speaker today, Dr. Richard Flavell. And we will also invite Richard to turn on his video and sound, and I let, Elina start.
Thank you, Carly, and thank you, Richard, for joining, and welcome everyone to the 2021 Global Immunotalks. So I could really not be more pleased and honored to introduce to you Dr. Richard Flavell, who I'm sure needs no introduction for most of you, as he's a giant in the field of immunology. So Richard is originally from England. He is currently a Sterling Professor of Immunobiology at Yale University School of Medicine and an investigator of the Howard Hughes Medical Institute. He receives his bachelor and PhD degree from the University of Hull in England and performed postdoctoral work in Amsterdam with Piet Borst and in Zurich with Charles Weisman. Before Richard accepted his current position at Yale, and that was in 1988, before that, Richard was first assistant professor at the University of Amsterdam, then head of the Laboratory of Gene Structure and Expression at the National Institute for Medical Research in Mill Hill, London. And subsequently, he was for 6 years President and Chief Scientific Officer of Biogen Research Corporation in Cambridge, Massachusetts. And so he was for 6 years in industry and then came back for a very successful career in Yale. So Richard is fellow of the Royal Society, a member of the National Academy of Sciences, and as well as the National Academy of Medicine in US. As you may imagine, he has received a long list of awards. It is indeed difficult to decide which one to mention, but this long list includes the American Association of Immunology In Vitrogen Meritorious Career Award in 2008 and the Vilcek Prize in Biomedical Sciences in 2013, again, among many, many other honors. So throughout his career, Richard has made many phenomenal discoveries that have really shaped in significant ways our current understanding on how the immune system works in both health and disease. So his approach is not a focused one to a particular aspect of the immune system or a particular immune cell type, but instead, Richard uses transgenic and gene-targeted mice to study both innate and adaptive immunity, T-cell tolerance and activation in immune responses, and also in the context of autoimmunity, apoptosis, and regulation of T-cell differentiation. During his PhD, Richard first co-discovered introns in cellular genes, showing that DNA methylation correlates inversely with and prevents gene expression. Later, as a postdoc with Weisman, he was the first to develop and employ reverse genetic, and then later in his own laboratory, he pioneered this approach, the reverse genetics in vivo, to study the molecular and cellular basis of the immune responses, an approach from which we all benefited in the field. With these and other approaches, Richard has worked and has been instrumental in discovering the molecular basis of T-cell differentiation from precursor cells into differentiated subsets, as well as the mechanisms of immune regulation that prevent autoimmunity and overaggressive responses to pathogens. Richard's team has also discovered the role of several receptor families within the innate immune responses, including toll-like receptors and intracellular node-like receptors. He has also established the connection between the inflammasome, microbial homeostasis and chronic diseases, and how this biosis of the microbiota can cause colitis and metabolic syndromes. And we could go on enumerating Richard's discoveries. Actually, I didn't even mention all his tremendous work with TGF-beta biology in immune regulation. But I think that we are all super eager to hear Richard's global immunotalk today, which is entitled A Humanized Mouse Model for COVID-19 Infection and Pathogenesis. And I heard that it's the first time he's presenting this on this topic. And so, Richard, thank you so much for joining, and thank you for all your efforts to prepare this talk and to present today.
Thanks, Alina, very much.
And so, as always, we like to know more and learn about the global immuno speakers, and there is so much we can learn from you, Richard, really. But many times we learn from mistakes, right? And so the question that we have for you today is, what is the mistake that taught you the most in your scientific career?
Sure, yeah. So the one I picked, it goes right back to the beginning when I started off as a young PI. So in those days, postdocs could be quite short because I did molecular biology, and I did 2 postdocs, but it was in less than 4 years, actually, combined, the 2 postdocs. Yeah. And so at the age of 28, I became a PI, and I thought I'm getting way too old, so I better get a job, so I'm getting anxious. And so I was at that time a very driven, scientist, but I was a dreadful micromanager, I think, which one tends to do. And initially I had young undergrads working with me, and that worked quite well because they need a lot of direction, so I was able to give that. But then I got my first grad student, and by this time, by the way, this was the first job which was at the University of Amsterdam, right? Mm-hmm. And of course, all trainees are different, right? And some are very thoughtful, and others are very fast moving. My first grad student was a very thoughtful and very careful worker, but at that stage of his career, he was a little bit slow. So I was way too hard on this poor student, who was a wonderful person, and became very successful, by the way. And so I was just awful. I was just an awful micromanager. I was way too hard on this student. But luckily, his brother, who was already a friend of mine, actually, was in the lab. And so I guess I was bad enough that at the end of one day, my grad student and his brother cornered me and took me into my office and basically read me the riot act. They said, this is ridiculous. I'm way too hard on this guy. This is not way to treat people, and they really, really laid into me. And so that was a real shock. This is not often you get something like that. But I thought about that, and I went home and I thought, they're absolutely right. And so what I learned from this, it was my first lesson in becoming a real mentor. And this really stuck with me forever. And so I think from that point on, I had a much better understanding of what our job really is in the lab, which is not only to get results, but to help people acquire, the skills and to enhance their abilities, to help them grow, et cetera, et cetera. And as I said, now let me emphasize, of course, I didn't get better overnight either. I was still sometimes pretty awful. It took me years, but nonetheless, I think that was a real epiphany. And I'm very grateful to both of them, and they're still very good friends. We're all very good friends, and I still see them from time to time. And both are very successful scientists, actually. And so that was probably the most useful mistake I made, I think, and it worked out okay in the end. And so I became very interested in being a mentor.
Perfect. Thank you so much, Richard. Thank you so much for sharing, the mistake and what you learned from that. I think it's very helpful for all of us to hear. And it is fortunate that was your first graduate student, that you learned the lesson very early, yeah. and how important also to listen and act on the feedback we get from mentees. I think that's
absolutely
that's key. Thank you, Richard. Okay, so without further ado, we should share your screen and enjoy your talk. Okay, yes, we can see the first slide, and then perfect.
Is that all there? Okay, great.
Yep.
Well, so when I, first of all, thanks very, very much for inviting me to do this. I mean, it's a great honor. I have to say I'm a huge fan of this series, and luckily I found out very early on, and I've made a point of watching every one of these, usually at the time, at this time, because it's right after some of our lab meetings. And it's been fabulous for me. I mean, because of COVID, I've been, like all of us, our lives have been disrupted. I've been working from home for 9 months or more, it was just wonderful. And what you have done, you organizers have done, is something really fantastic, which is to link us and the whole world. And you've created a series with enormous diversity of speakers, which is just, I mean, it's absolutely fantastic. So then of course, when I wondered what I should talk about, I thought that this project on COVID surely is, for us, is maybe the best thing to talk about, because it's the reason we're all here. And it's caused so much misery, death, it's a terrible scourge, but we're here with it. And what we decided in the lab is that we should try and do something about it, like many workers and many great scientists have contributed to this field already. And so a young postdoctoral fellow, Esen Sefik, originally from Turkey, and then trained with Diana Mathis and Christophe Benoit in Boston, became interested in this. She was already becoming expert in our Humanized Mouse project, and we decided, and she decided, to work on this to see if we could develop an infection model. And the goal, of course, was to create a model for COVID. Now, of course, that's a complicated matter because it's a very diverse disease. But as you probably know, those of you who follow the modeling of the disease, there are many models, but most of them model probably what I hope happens most in people, which is an acute viral infection, which is pretty asymptomatic and goes away. And what we were interested to do is to create a model which basically addresses the severe cases where you have chronic severe disease in people. And this is where, of course, the terrible morbidity and mortality comes from. So now first I should explain what we mean by a humanized mouse. So there's a variety of different uses of this term. In some cases, people mean putting human genes in mice in order to study the functioning of their genes. But what I'm going to be talking about is putting human cells into mice and studying the effect of the human immune system itself in this mouse environment. And this is the mouse that I'll be talking about. We call it MISTRG6, and it's turned out to be a very useful model. So I'll first explain what we're talking about. So I was stimulated to want to do this project when I saw a paper from Marcus Mann's lab, which you can see at the bottom was in Science in 2004. And what Marcus had done is taken previous work, which had developed humanized mice, and developed a very nice model in which he could take a variety of human hematopoietic stem cells. He took them from cord blood, and then to inject them into an immunocompromised mouse. That was done in baby mice, right, mice just after birth, and by injection into the liver, which is of course a hematopoietic organ at the time. And if you do that, and as Marcus and his colleagues showed, you get mice in which the human immune cells derived from those progenitors develop a lot of different immune cells. And in this nice paper, he showed you could get B cells, T cells, and a lot of different cell types. Now that's that's part of the series of kinds of humanized mice of this type, meaning the cells in mice that were developed. And it goes back all the way to the use of skid mice, which were used first. Now, of course, the reason for that is that human cells would be normally, and are normally, in an immunocompetent mouse rejected. And so introducing the skid mutation reduced that. And then the next breakthrough was really to introduce the Rag gamma common chain mutation, which eliminates NK cells because it takes out IL-15 signaling. And that was a very important improvement. And Marcus did that with his model there. And the similar work was done on the NOD background, which is what you can see here. And let's use the laser pointer. And these are various models that existed. All of them, as you can see, bear very little, well, have some things in common with human. You can see an example here of blood and bone marrow there. But there's a lot of mouse cells of the immune system which are still present, and the distribution of the cells wasn't and has still been very aberrant. And it occurred to us that of course, in a way, just to expect the mouse to be able to support the immune cells of another species is a tall order. And that what we needed to do really is to manipulate the microenvironment. So what I decided to do is to try and combine the cellular humanization with molecular humanization of human genes. And in doing that, we teamed up with a group of really talented people at Regeneron Pharmaceuticals that had worked out a really excellent pipeline of genetic humanization. And the approach that we followed is schematically shown on this slide, where you take a mouse gene, which you can see here, and what you're going to do, or what we did, is to insert the human corresponding gene into the mouse locus. And then you end up with the human gene, but it's in the environment of the mouse gene. And obviously, the goal there was to have a gene which you would express at the right time, in the right place, and at the right amount that is made in the human. And the goal, of course, was to have this occur in the microenvironment in which the human immune cells are going to develop. And this is from a slide from Tim Villinger, one of the people in the lab who did that work at the time with us. And this worked really well, I should say. It worked in a modular fashion. We made, as you can see, multiple molecular substitutions. Each of these, this MISTRG is an acronym for the components we did. So we humanized human MCSF or CSF1. That provided myeloid cells, monocytes and macrophages, macrophages in the tissue, and monocytes in circulation. We humanized IL-3 and GM-CSF, two co-localized genes. And GM-CSF is required for alveolar macrophages. And that, I should say, each of these things that we did, fortunately, really worked. They in a modular fashion provided the cells to which they were feeding basically as growth factors. So we got macrophages from these two guys. We got long-term HSC maintenance from thrombopoietin. We introduced SERP alpha initially after a conversation with Irv Weissman, a wonderful guy. Irv pointed us to this one, and of course human SERP alpha we humanized. And that provided phagocytic tolerance. So the mouse macrophages no longer ate and destroyed the human cells quite as much. And these two genes, of course, were to create an immunodeficiency that I'd already mentioned to you. And sure enough, I mean, this, don't look at these details here, but the principles are that certain factors are required for each of these lineages, and that actually worked quite very well. Well, after we did them individually, making mice having number one or number two, etc., we combined them. The we in this case was Anthony Renvaux, a wonderful postdoctoral fellow from Belgium, who's now a faculty member at the Hutch in Seattle, and Tim Willinger, who is now at the Karolinska Institute, also with his own lab. Combined these genes. SERP alpha work was done by Till Strovig, who now has his own lab in Berlin, in Germany, and so on. And luckily, this combination worked very, very well, and they just worked well together and made an even better system. And that's what we call this MISTRG mouse. And by the way, these animals are almost at JAX now in their newer form. And these will be available very soon, very easily to the whole scientific community. So looking at this, at these mice, the first thing that we were really interested was to get functional myeloid cells, because they're so important in many aspects of the immune system. And as you can see here, this worked very well. This is just an old slide from a paper that we published in 2014. And you can see here that this is looking at blood monocytes, basically, and you can see in the older systems there are very few circulating monocytes, but with this incorporation of the CSF1, you get a huge improvement. Each of these dots is a mouse, so there's a huge number of animals that we analyzed. The tissue macrophages, you can see here just by staining with CD68, showed human macrophages in all the tissues we've looked at, basically lung, liver, in the intestine, and so on. And a second benefit came from these myeloid cells because they actually provide interleukin-15, which gave us NK cells, which it's a little bit hard to see here, but that's staining with NKp46. So the mice worked very nicely, and Anthony and Tim used them for various purposes there. The immune cells are functional. They respond to bacterial infection, as you can see here, or to sepsis. And so it gave us a nice system. The monocytes that you get in these mice are very similar to human monocytes in blood. And as you can see, comparing CD16 and CD14, you have these three populations, CD14 single positives to CD16 single positives. And sure enough, when we looked in the mice, this is comparing these MISTRG mice. This shows blood, which has those three populations of human monocytes. And in particular, particularly relevant for today's talk, actually, these CD16 single positives, which play such an important role in infected lung, for example. And so this gave us confidence that we could hopefully use this system. We added finally interleukin-6. That was done for a couple of reasons, but that's helped in the context of today. What it does is to act synergistically in early hematopoiesis. And so it makes the system very sensitive to cell number, meaning you can put very few cells in and get very good results. So it helps, for example, when you want to do experiments with patient bone marrow. And finally, if you do single cell sequencing, you can find all these populations, and these mice, as I said, really have a very good distribution of most human cells. And in particular, you can see the macrophages and monocytes in this particular annotated single cell experiment that we did with Avi Regev and her colleagues. So we decided to go on then and assay and analyze SARS-CoV-2 infection. And to do this, we were able to team up with Akiko Iwasaki at Yale, and Akiko had worked out this very nice way of introducing the receptor for the virus, human ACE2, using an AAV vector. And this vector was provided intratracheally, and that gets it into the lungs of the mice. And here you can see staining that shows that ACE2 is expressed, and that SARS virus, actually, the CoV-2 can infect these mice. And Ben Israelov and Eric Song in her lab did just a super job with this, and their paper was published middle of last year. And so Eric and Ben, particularly Ben, helped Essen enormously getting this project off the ground. And Essen then learned the tricks of the trade And we use this to put the receptor into the lungs of MISTRG mice. And here's the protocol we basically use. So here's humanized mice. Remember, we give them the human cells almost at birth. We let them get 8 weeks old, and by that time the immune system has grown up very nicely. We then infect them with the AAV virus to give them the receptor. 14 days later, we infect with the virus, and then we followed infection. Now, the first thing we've noticed is that in contrast to most of the other models, this turned out to be a very nice chronic model, which I'll show you on the next slide. But so basically we could follow out infection out to a month and probably longer. We just haven't gone further than a month yet, just because of the obvious reasons. And so this slide shows that these mice sustain a very high titer of the virus and viral RNA. And it'll enable me, I'll show a comparison of a normal mouse with, this is a B6 mouse, for example, which only has mouse immune cells, and confirming what Akiko and others have shown, you have an acute viral infection, which then gets cleared by a week. However, if you look at the bottom, or look at all the way across on the right-hand side, where hopefully you can see my pointer, that this infection is maintained for a longer time. And if you look at the RNA of the virus, we have viral RNA and components going out for a month. And so this is very different to the other models, and a lot more similar to what's happening in severe patients, where you have a chronic rather than acute situation. Note that this chronicity requires the presence of course, the ACE2 receptor, but also the human cells. If you don't have the human cells, you don't actually get large numbers of, you don't have this long-standing infection. So the human immune cells are actually contributing to the pathogenesis in interesting ways. Now these mice get sick, but they, and now on this slide, everything is a MISTRG mouse. And we're just comparing mice that have human immune cells, were humanized, in other words, got the receptor and were infected. And the bottom line is that group of mice gets sick, and these mice lose weight and they can never gain it back, at least over a month. That's in contrast to all the other kinds of mice here, which are either uninfected mice, all of this, of course, being in BSL3, and the mice which have human immune cells but not the receptor. These mice all grow healthily in the system, but the mice that get the virus, they lose weight and they're chronically sick, actually. And this shows that, of course, there's great heterogeneity in the system in terms of exactly what one animal does, because of the individual differences in humanization, in viral load, etc., etc. But basically, as a group, they get sick and they stay sick. So what's going on in these animals? Well, first of all, if you look at the histology of lungs, they get an terrible lung inflammation. You can see this is two days through to a whole month, and this inflammation in the lungs stays basically all the way through the month, and this is uninfected, obviously. And if you look in detail, you can see this, even at day 28, you've got these really inflamed areas. The other thing that happens is by day 28, we start to see signs of fibrosis. And I can show you, this is using a trichrome stain for collagen. You can see that these lungs now are becoming fibrotic, although this is still early in the fibrotic process, because they are still inflamed, and we need to go a longer time point to follow this a bit more precisely. But that's clear from those studies. So what is happening during this time period? Well, if you look at the numbers of immune cells in the lung, an uninfected mouse, which is you see in white here, of course, has very few immune cells in the lung. But as soon as 2 days after infection, there's a flood of human immune cells that come into the lung. They increase. There's a slight dip, perhaps, at 14 days, but then off they go again. And if you look in the bowel, and this is the bronchiolar lavage, which is this is cells which extravasate out of the which leave the alveoli and enter the space itself where the extracellular fluids are, you can see there are almost no cells in that space in an uninfected animal, but very large numbers of immune cells from day 2 all the way up to a month. What are these cells? Well, this is showing the percentiles of various cell types in the lung. So of course it's deceptive because uninfected, there's very few cells. So you have to correct for that. But one thing that was very obvious very early on is that there's a big influx of monocytes of all 3 varieties. And at first you have alveolar macrophages, but subsequent to that, we get a lot of inflammatory macrophages. And you can see that better on these slides where you can distinguish what is what. This shows macrophage numbers. And again, you see this big increase around day 7. There's a really huge number of human inflammatory macrophages, as I'll show you in a minute. Whereas, of course, here, these are alveolar macrophages. And basically, if you look at the specific cell types, you can see alveolar macrophages get diluted out as a percentage, and inflammatory macrophages become the majority of the cells, and as the process proceeds. T cells get activated in the lung very fast, actually, and as early as day 2 and day 4, we see T cell activation, and as I'll show you, this appears to be two responses. The first is gamma delta cells in the lung, human gamma deltas. Of course, I'm talking about human cells all the time now, unless I say otherwise. And you can see using markers for activation like HLA class 2 and CXCR3, which is a marker, of course, for inflammatory cells like gamma producers, this kind of thing. You can see there's plenty of these activated cells really right in the beginning. And if you look at what these cells are, first of all, as I said, you see gamma deltas come up very early on. And the second thing that we see is bystander activated CD4 cells, which presumably are some form of activated memory cell. Human cells are well known to be able to respond to cytokine activation. In other words, cytokine signals lead them to produce cytokines. And you can see here that if you do, looking at the bystander activation genes in these T cells, a huge amount of this response actually are interferon activated, interferon responsive genes. So that is the early T cell event which happens in these animals. Now, just as in human patients, these mice develop systemic lymphopenia. So in this slide, which is a bit busy, I'm sorry, shows the humanization, the number of T cells in each mouse at the start before they get infected. Then you see what happens to them. Obviously, you can only sacrifice a mouse at one time. So you only get a dot going from here to there or from here to here. You don't get both. But you can still see by analysis that you get lymphopenia from day 2 onwards. There's a blip going up by day 7. We'll talk about that maybe in a bit, and then back down again, and so on. And the same happens in spleen, by the way. So this is which you would expect. Spleen and blood being, of course, reflecting similar things. So this observation that we see in human patients seems to happen in these animals. And this is a flood of T cells, I'm sure, going into the lung in response to the infection. So Essen took a look at the transcriptome in these animals to determine what's going on both at the whole tissue and single cell level. And the most striking thing is, as you might expect, that you get an enormous interferon response in these animals, and 800 of the 1,000 genes which are looked at actually are interferon responsive genes, both, by the way, in this case in the human cells and also in the murine cells, because you can look at that in total RNA sequencing separately. And you can see there's evolution in what the interferon responsive genes are as a function of time, but at all times there are these interferon responsive genes. Now, looking at we did single cell sequencing at day 4, and we're doing many more, but it's day 4. What you can see is that these populations of cells, I hope you can see this up there. this at day 4 of lung cells shows these are monocytes and macrophages. These cells, cluster 5, are alveolar macrophages, and these are the T cells here. And I think what you can see by this very clustering analysis very clearly that the inflammatory cytokines are mostly being made by these monocytes and macrophages. And here's a lot of IL-1 beta, IL-18, et cetera, et cetera. And interferon gamma, of course, is being made by the T cells, cluster 4, you can see here, and so on and so on. So there's a lot of inflammatory cytokine production as seen in human patients. So from this first level analysis, it shows that these mice, when they're infected, get high viral titers, sustained viral RNA production. They show immunopathology in the sense of infiltrating activated immune cells. They begin to show signs of fibrosis after about 28 days, sometime between 40 and 28 days, say. Monocytes and monocyte-derived macrophages infiltrate the lungs early, and they make pro-inflammatory cytokines. You get T cell lymphopenia, as you see in patients, and a sustained interferon response for a month. So we thought it would be worthwhile to use these animals to study infection. And so we decided to look first at antibody responses to see to what degree antibodies impact the infection. So we teamed up with Michelle Nussenzweig's lab, and Michelle had done a really beautiful job of looking at of course, early in the pandemic, New York was a hotbed of COVID, and so they were able to analyze a vast number of patients in this way for these antibodies, like 150 patients. They collected plasma about 40 days after symptomatic onset, and characterized those antibodies. And they showed, that on average, the people have low antibody titers, but quite a high levels of neutralizing titers in some cases, 80% of which was directed against the RBD and 70% against the S. And so we took the convalescent serum that Michelle had, a pooled convalescent serum he kindly provided to us, and we tested whether this convalescent serum is able to prevent infection. And so this is our protocol again. Remember, we engraft, we let the mice get a good immune system. We give them the AAV-ACE2 that Akiko developed, put that in, and then 8 hours before we are going to infect, we give them the convalescent serum. As you can see here, a single dose, so 100 microliters. And then we infect, and you can see that this immediately has an effect on viral titers. These are PFUs measured by plaque assay, and so you can see here untitered. You've got an obvious reduction by the serum, but it doesn't blow away all of the virus, and as a result, it does not prevent the human infiltrate. As you can see here, the human macrophages still go on to be a major presence there at day 4. So it's a partial response, but that led us to think it would make sense to use some of Michel's really excellent neutralizing antibodies, and he recommended and sent to us these 2 clones, which he had, of course, cloned antibodies. These were both spike antibodies. And they were, but against different binding sites, complementary binding sites, actually, both with high neutralizing activity. And so, again, we used them first in a prophylactic mode. And as you can see here, that we gave them at the same time 8 hours. Here's the dosing, etc. And basically, in prophylactic mode, these antibodies work. And this shows that we can block the weight loss that these mice develop. In this case, we use these antibodies separately. It's a very similar story. One is slightly better than the other. I'm not sure what that means at the moment, but that's not, I think, level of resolution beyond which we were interested to look at that stage. And sure enough, if you look at the virus at day 4 again, So this is now basically 4 days after we treated the mice. You can see that the viral titers are really knocked down by at least 4 logs, as you can see there. And the viral RNA is knocked down by a similar amount. So that was good. The immune infiltrates are indeed reduced. And if you look at BAL, I mean, which is a reflection, I think, of the response to and damage to the lungs and so on, you can see that these antibodies actually are both very effective at preventing the immune cells in the bronchial alveolar fluid. The macrophages that develop normally in untreated animal by day 4 already, they're pretty much gone. And the same is true, even more strikingly, actually, in the bronchial lavage. So prophylactic use of these antibodies is really very effective at preventing infection. They also prevent, by the way, the systemic lymphopenia which develops, presumably because the infection is blocked. And you can see here, the splenic T cells are not reduced like you get in here, nor the blood. So then we wanted to move on and try them in therapeutic mode. Of course, this has been partially successful, but limited, I think, in the clinical experience, such as it is. That's still being, I guess, partially evaluated in different environments. But the plan, I mean, I think the message from that is you have to give the antibodies early. So Michelle recommended we do, an early time point and then, a somewhat later time point. So what we did is we gave the first antibody at day 11 hours after infection. Crazy time, but our animal lights go off, and so we had to work with that. In BSL-3, you don't get to make the rules. So 11 hours or 35 hours, we gave the antibody. These are the doses, single dose IV, but this basically has not been successful. Even as early as 11 hours, the weight loss that we observe under these conditions is not blocked. Now, I should add that we use a lot of virus. I didn't mention that, but it's 60 million doses of virus. So it's not impossible there would have been an effect if the disease course was allowed to be slower with a much lower inoculum. But that's what we see under these circumstances. However, the viruses do neutralize. Sorry, the antibodies do neutralize the virus, at least again, in this assay, at least to the level of 3 logs. And so they are very effective at mopping up virus, but they don't block pathology. Immune infiltration continues. This shows, again, this is cells in the bowel. The early treatment was partially successful at preventing the bowel, presumably a result of its early intervention in the viral infection. But the monocytic infiltration, which comes into the lung, is unaffected. Monocytic infiltration still occurs in the bowel. Again, early there's some improvement. So there's some activity of the system, and so even though the mice are losing weight, they've lost a lot of virus, and they are responding less aggressively, apparently, to the virus. So in a way, we've got a separation here of immunopathology from a viral load. The macrophage infiltration, which occurs in these mice, is, as you can see here, is basically in the lung tissue itself is still occurring in both cases. Although, as I said, in the bowel, we are able to reduce those numbers. So the conclusion for the first part of the therapeutic intervention is that monoclonal antibodies give results, I think, pretty similar to what is seen in the human experience. They're good at neutralizing the antibodies. They work prophylactically, and when given very early, they show some utility. Prophylactic treatment really works, but a therapeutic efficacy is limited unless really early administration occurs in our system. Of course, you have to correct that for in one's thinking about the dose of virus and the stage of infection. Now, just to conclude on therapy, the one thing that I think has had an enormous impact on COVID therapy and has really distinguished early disease and mortality from what we have these days is treatment with steroids, in particular dexamethasone. Of course, dexamethasone is a very potent immunosuppressive steroid, and it's used really big time in the clinic now with serious COVID infection. And so we decided to test this in our model, again, with the goal, I mean, to see to what degree we are reflecting what happens in the patient, which is what we're hoping, of course. And so we decided on a protocol for dexamethasone to mimic treatment of patients which to a degree would be similar to the patients which are already have established long-term infection, clear pathology, and probably attenuated to a degree a viral population. And so we treated them with 3 days in a row, day 7 through 9 after infection. Remember, we infect here you know, 2 weeks after we give AAV and the receptor. And one thing, and you can see on the right-hand side of this slide, immediately you can see it works. The untreated mice undergo this pattern of loss of weight, which they never get back. But if you treat at day 7, and this is upon treatment here, you get an immediate improvement in body mass, and that's maintained. So the parameter we use for quote health of the animal shows an immediate response, and this weight loss, I think, of course, such weight loss is also observed in human patients. The late dexamethasone reduces the immune infiltration into the bowel and the lung tissue itself. Note this is an expanded scale here. So we knock down a lot of the infiltration. And I should emphasize, we're measuring at 2 weeks here. We're going on to measure 4 weeks time points now. So the question is, what's going on here? And what's happening on a cellular basis? Well, on a cellular basis, you can see that the composition of the cells that have been treated with dexamethasone are very different. The numbers of macrophages are really knocked down. You can see here, this is macrophage numbers. That's percentile on the left, of course. You can see that in the control, you have a lot of macrophages in the lung. They're basically whacked down by the dexamethasone. The same is true, by the way, for the monocytes. We'll have a look at that in a second. The second point is that T cell activation, which occurs in the infected animals, is immediately attenuated, and you can see here, well, immediately, but by the time we measure it, obviously. And so by measuring class 2, you can see this is the control, which is infected. The levels of class 2 on the T cells in the lung at day 14 are reduced, as is the expression of ICOS, looking at a T cell activation marker. So it's immunosuppressive for both T cells and myeloid cells. Now, the question is, what happens if you give dexamethasone closer to the time of infection? And that actually turns out to be a disaster, which, of course, one might imagine. And so here you see, instead of the adding the dexamethasone after a week after infection, we give it 3 days after infection for 3 days. And in that case, actually, you get a catastrophic decline in the animals. And by day 7, these animals were moribund. They had to be sacrificed, otherwise they would have died, of course. And you can see there's a catastrophic weight loss which occurs. It's even worse than what is seen in the normal animals. And this is a consequence of the elimination of the antiviral response. And you can see here, if you look at, I'm sorry, This is looking at the human immune cells, first of all, in the lungs. These human immune cells, are, of course, are completely eliminated, but obviously the immune cells themselves are important for resistance to infection. And this you can see very easy by measuring viral titer, where you can see if you treat with dexamethasone 3 days after infection, you get a 3 log increase in viral infection within a couple of days. So this is of course what presumably the animals are dying from this catastrophic viral infection, and the viral RNA and the viral titers mimic each other. So on dexamethasone, I think what we can conclude is that Dex reverses immune activation in macrophages, T cells, and neutrophils, and that is curative actually for animals late in infection, as mimicked by these various parameters. And we can block weight loss and we can block immunopathology. But the early antiviral response is essential for viral control, and therefore it would be very important in the clinic to be really cautious about not adding dexamethasone too early, as one hears anecdotally from clinical experimentation. So what can this system now be used for? I think our purpose with what I've shown you up to now is to give a sufficient information, I think, that suggests that at least for one kind of COVID infection, this is a pretty decent model, because it mimics it mimics chronic infection, chronic immunopathology, and even probably fibrosis, which is happening at later time points. And so what are we doing now? Well, now we really want to get into the driving causal mechanisms of the disease. I think we've shown enough to say that we can assess new therapeutics because we can validate old therapeutics, and we could use these mice, of course, very nicely to ascertain how well vaccine approaches work, because one can take, of course, vaccine materials and apply them to the animals. So let me then summarize in closing what we've seen. And I'm sorry, this is an awfully busy slide, and I won't go over every point. But I think the point of this first of these two slides is that I think these humanized animals infected with COVID-2 do recapitulate a lot of chronic human disease features. The mice lose body mass. The humans of course do the same. The duration of disease pathology is long rather than acute. It's a chronic disease. We don't get mortality unless we suppress the antiviral response. That's not, of course, in humans, as you very well know, but mortality is a different matter. And so that's an issue that needs to be further worked out in our system. The lung pathology looks pretty similar according to our pathologist. Now, all that pathology was monitored by a board certified pathologist. So there seems to be quite a bit of similarity. And of course, the fibrosis signs that we've seen are interesting. We'll follow up on that. Lymphopenia occurs in both. Macrophage responses are very similar. The chemokines and cytokines are extremely similar between the two species. Well, because the cells are the same species between the model and the real disease. T cell responses are interesting and similar. We don't know much about the specificity of the adaptive response. I'm sorry, I didn't get into that very much. But I talked about the first wave of infection, but there's a second adaptive wave of infection, which comes up around between day 7 and day 14, as you might expect. The interferon signature in both cases is sustained for a long time. And then finally, we are able to get similar results with therapeutics. And then if we compare our model to other animal models, I think our model is much more a model of the chronic disease in humans, whereas I think pretty much all of the other models are more a reflection of acute viral infection. And of course, both things are useful. both of these things happen in patients. And we're hoping, of course, to exploit the chronic features in order to learn about how chronic disease happens as much as we can, predict things that could be useful in the clinic, hopefully, and complement the work of the clinicians on the one hand and the people studying acute disease on the other. So I think I should close here. It's already getting late. And thank everybody. That this is Essen. Essen deserves a huge percent of the credit here. Essentially everything I showed, she did almost single-handed. She was helped in the very beginning by Ben and Eric, particularly Ben stuck with her and helped her with titrating virus, giving viral stocks, and initially doing the infections and so on. Kiko, of course, many thanks for allowing us to use her model and to apply it into our system. Craig Weiland, Craig's the virologist, obviously, and he gave us virus, and he's given us a lot of great advice and collaboration on that. A lot of bioinformatic work was involved in this, and we have a long-standing collaboration with Yuval Kluger, and we actually co-supervise grad students, Jun, Zhao, and Rihao, and now Wes. These are our co-supervised students who helped enormously with the analysis. And Michelle provided and collaborated with us on the antibodies. Michelle gave us these fabulous reagents, and we had a good advice, of course, on how to apply them. And the whole lab, of course, interacted in various ways and gave lots of feedback. This is a lab get together. We have a retreat every year, and this was a tie-dye fest, and so you can see what it does to people's behavior. And then finally, I want to thank the people that got the whole thing going. This goes back a long time. We started to make these mice with the funding from the Bill and Melinda Gates Foundation, very generous funding for a long time. It all started when I saw Marcus's paper. This is Marcus right here, Marcus Manns. I saw his paper. I thought, oh my goodness, we've got to collaborate with him and combine our genetic engineering with cellular engineering. And this turned out to work very nicely. And we worked with Marcus for a long time. Marcus is now head of hematology at the University of Zurich. At the time we worked together first, he was in Bellinzona in southern Switzerland, Ticino. And many wonderful people worked with us. In my lab, Anthony Ronvaux, he's here. He's characteristically modest. He did the lion's share of the MISTRG work in that paper together with Tim Willinger. As I said, Anthony is now at the Hutch. Tim is now at the Karolinska. Till Strovik is here. Till is in Berlin now. And we collaborated with Regeneron, a lot of years of work with them, supported by the Gates. And Sean Stevens was the science person there. And Kagan Gura then took over from him. This is Kagan. This is an old slide, actually. But these are, many people have been our collaborators. And Bill Philbrick has been involved in a lot of the genetic engineering sides in recent years. And I should thank Elizabeth Einem, who really got us, looked after us all, got all, lots of very good scientific input and so on. So thank you very much, Elena. And I'm a big fan of your Global Immunotalks, and I'm very honored you let me speak in it. Thank you.
Richard, thank you. This was wonderful talk, and what a valuable tool and terrific insights for a very important problem. So thank you so much. As always, we do the questions via Twitter. So Carly will explain with this last slide how to ask Richard's questions via Twitter.
Yeah, thank you, Elena, and thank you, Richard, in particular, for sharing all this data fresh from the BSL-3, so very much appreciated. So as a reminder, there's already a tweet that you can find in our Twitter account. So please, you can go to the Global Immunotalks Twitter account. You'll find a tweet saying, ask questions for Dr. Richard Flavell here. Make sure you reply to that tweet, and do not forget to mention the hashtag Global Immuno, and Richard will be making use of the ImmunoSpeaker account to answer your questions. So feel free to ask any questions for this fabulous talk, and thanks again, Richard, and we look forward to being together with all of you next week with Dario Zamboni, who will be joining us from Brazil. Thank you so much.
Thanks.
Thank you. Thank you, Richard, again, and bye, everyone. See you next week.
See you.
Bye-bye.